So welcome to our lesson. We would like to talk today about the degenerating brain. And I would like you to stop me whenever anything is unclear, because this topic depends on some understanding of the molecular biology terms. So if there's any question, raise your hands and ask. When we talk about neurodegeneration, we of course mention Alois Alzheimer, who about 100 years ago identified a patient who was 51 but looked like 80 and lost the capacity to remember anything. She only knew her first name. And he was a neurologist. And the story I heard is that when he moved in Germany, if you want to get promoted, you are moved to another city. And he moved with a jar with the brain of this patient who died at 51. And he made very interesting sections and drew. He was a very talented artist. I'll show you some pictures of the brain cells that he drew. And until today, the disease is called Alzheimer's disease. There was an effort last year of the Israel Academy of Language to Hebraicize the name of the disease. And there was a big discussion about that. And of course, you can't Hebraicize a name. So eventually they gave up. And we still call it Alzheimer's disease. What I'd like to tell you today is that we have new hope thanks to research efforts, multi-leveled in many countries, and I'd like to discuss that with you. We will mention a new family of genes, microRNAs, which break all the rules because they don't do what the genes, the classical genes that we grew up on, do. They don't produce any protein. Instead, they wrap around another gene product and stop it from being expressed. So instead of a plus activity, they do a minus one. They function in direct association with our genome. So people with genes that do not communicate well with the microRNAs, which I hope to show you data for, would show a different phenotype. They'll have different properties. They'll have different patterns of genes in their brain. Last but not least, we believe that all of these genomic changes affect the network of neurons in our brain. So here we go, Alzheimer's disease as it looks from the researcher's point of view today. How bad is it? We live in a world where life expectancy is extended on a yearly basis. Now you have to realize that 20 years ago when we said life expectancy is prolonged, what we meant really was that babies and young children stop dying. 2 generations ago, many children who were not strong enough either died as babies or before they reached the age of 5 years. When we calculate life expectancy, someone like that lost, let's say, 75 years. But as soon as we protect children and we protect babies and there's no childbirth death and much, much fewer deaths of young children, then the average life expectancy goes up. Today we mean something else. Over the past 20 years, babies and young children don't die. What we mean to say when we talk about life expectancy is that people age more. So people live to 100 or more years old, and this is something we didn't know before. Now, Alzheimer's disease, the major risk is age. That means that in a young population with a short life expectancy, you will not see Alzheimer's disease. And in the 60s of the last centuries, the Japanese researchers insisted there is no Alzheimer's disease in Japan. Indeed, there wasn't, because the life expectancy in Japan was 56 in average. People were dying because of esophagus and intestinal tract tumors, because they were polishing rice on asbestos, which is carcinogenic. when that was discovered and they shifted the way of production of rice, life expectancy in Japan got prolonged by 25 years on average. Today it is 85, and they have a problem of Alzheimer's, definitely. Last time I visited Japan, in the hotel, in the drawers you could find diapers for aged individuals because it's a major issue. So aging world means that we increase the risk of neurodegenerative disease, not for a bad reason, for a good reason, because they reach the age of diseases for which the risk factor is age. So today we talk on average of 1 in 8 of people above 65 years of age, and these are the predicted costs, which is frightening. So the entire world realizes that it's time to do something and do it fast to solve the problem of Alzheimer's disease. How do you do that? You can, yeah.
Sorry, these are the costs for dealing with the people that have Alzheimer's?
No, this is the cost to society, because you need to take care of someone like that in 3 people per 24 hours. You need to take care of them around the clock. Either you have 3 people working for you at home, or you move them to a protected living where there are 3 people taking care of them. So the cost to society is immense. So how can you fight an important issue like that? One way is to develop new technologies. We'll talk about that. The other way is to change the way we understand things. We'll talk about that too. And the third aspect is to find new genes that we didn't know about, and we'll talk about that as well. So, Alzheimer's disease, how many of you have seen this movie? Okay, so you know it becomes an issue also at the cultural level. If someone in Hollywood thought that it's time to do a movie on Alzheimer's disease, that means that at the sociological point of view, this became an important issue. And she's a great artist. So, but what the movie shows is something that is much nicer than the real disease, I'm sorry to say. Okay, and how does the disease look? So these are sections from brains of people with or without Alzheimer's disease, same age. And what you see first and foremost is the loss of brain mass. You see enlarged chambers in the brain over here. You see folds that become deeper. You see that much less space is occupied by brain matter. And if you stain the sections, you can see that there are brown spots. These are the plaques that Alzheimer's detected. So we still use the same type of staining that he developed 100 years ago with a very poor microscope. So that's the way it looks at the level of neuropathology. But is Alzheimer's disease an issue of neuropathology? If you were asked, you know, what's the major hallmarks of Alzheimer's disease, what would you say? I mean, we don't see what happens in the head of people. What is the major hallmark of Alzheimer's disease? Yeah, loss of memory. First short-term memory, then also long-term memory. So the major aspect is cognition. In that very first patient, both aspects were met, but it is not that way for all patients. There are people with Alzheimer's plaques who do not show a cognitive decline. We still don't know why. I have a friend in Chicago who shows slides of a brain of a lady who died at the age of 91, and then he shows a poem that she wrote one day before. She was cognitive and alert, and the brain was full of Alzheimer's disease plaques. So we need to remember that there are 2 aspects of the disease, cognition and neuropathology, and that not always both of those appear. So how do you protect from that? Another aspect that I need to disillusion you about, people used to say until not too long ago that the brain keeps nerve cells until the age of 20, and then from 20 years on, you lose many thousands of nerve cells per day, and that's about it. Not true. Neurons in the human brain continue dividing to a very old age, actually. The last article I read was culturing neurons from brains of people who were 75 years old, and the nerve cells continued to divide. The issue is that you need more than that to maintain them in an active way. That is, suppose we take a brain region, the hippocampus, and we look at it enlarged. So here are the progenitors, the cells that will divide, and they keep dividing, and then they grow processes through which nerve cells talk to each other, and only if they talk to each other they survive. A nerve cell needs information. This is the material that keeps it alive. That means that suppose you stay in bed and you don't do anything active with your brain, neurons will divide and die. They'll divide and die. You need to keep cognitively alert to keep the neurons alive. Or as my one of other friends of mine says, use it or lose it. You need to keep brain activity to maintain the network functioning and protect the new nerve cells that keep getting born. Okay, so here is where the hippocampus is. This is the region where we always count neurons more easily because it's well organized. As you could see here, there are layers that are packed with neurons. So in an average rat, we know that there are 4,000 nerve cells that get born in this particular region of the brain every month. And in humans, there's no quantitation because the material is much more rare, but definitely we do have neurons that keep getting born. How do you study that? So brain research has gone through a lot of revolutions in the past decade or so. This is a new paper. It's from 2012. So 3 years ago, people realized that the brain of Albert Einstein is kept somewhere. He donated it to research. And they analyzed the structure of the brain, and they published an article in a very well-known journal called Brain, surprise, surprise. And they thought that Einstein's brain has special features to it. This is very old-fashioned research. Nobody would do such a project on a regular brain. But what we can do, so the concept on which this paper is based is that this is a very special person. He has a special brain. Therefore, it's worth investing the effort, even if all we can do is look at the contours. And that reminded me a quotation from Tolstoy that says, all happy families are the same, but each unhappy family is unhappy in its own way. And I'd like to turn the argument that on which this article on Einstein's brain was based and say, actually, we think the opposite. Like, if there is a disease, then all diseased brains should share similar features, whereas the healthy brains might be different. So let's see if we can do that. Of course, this was the case because Einstein won the Nobel Prize and his brain is considered very important. So what can research do today that couldn't before? We can put a person in a machine and look at the activity of their brain. So this is the activity of the Grateful Dead drummer, Mickey Hart. And what you see here is that different regions in his brain show activity at different times during the time that he plays the music. So different regions in our brain are involved in creative activities. Okay. And this is certainly something that we can't do for Einstein's brain. But we need to remember that we have today the capacity to look into the brain of live people. That gives us a new power. So what was Alzheimer's disease all about? One of the first things that was found in the Alzheimer brain is that one neurotransmission pathway is the first one to be damaged. So what do I mean when I say a neurotransmission pathway? I mean communication between nerve cells through a chemical. Now, this by itself is not a simple issue. Until about 100 years ago, neurobiologists believed that nerve cells communicate by sending sparks of electrical signals to each other. And what I'd like to tell you about is this gentleman, Otto Levy.
How the nerve transmits its signals.
This was the person who initiated the concept of information being transferred. This is the guy. And let me stop that. Okay. So why did I bring that up? Not because it is the Holocaust Day, but it's highly relevant. About 10 years ago, I was invited to Marburg in Germany, where a conference was held to celebrate 100 years to the discovery for which Otto Levy won the Nobel Prize. And his idea was very new at the time, so he made the discovery in 1906. He worked in Marburg, and he wrote it in his autobiography. And at that time, great scientists believed that they need to tie their discovery to a brilliant dream. And he's not the only one. I know about several of those who told the story as if they found it in a dream. And what Otto Levy wrote in his autobiography is that he believed that neurons can communicate through chemicals. And he had a dream, and in the dream he found out how he can prove this. And then he woke up and he forgot his dream. He knew he dreamt it, but he forgot the pathway, and he was very upset all day long after. And then the next night he again had that dream, and then he woke up, he got dressed, he went to the lab, and he writes he did something no distinguished German professor would have done. Anyway, he performed a real experiment in his own hands. You have to realize that this is important because this is one of the issues on the basis of which the Nobel Committee decided whether someone deserves a prize. So it was important to him to say, I did the experiment myself. And what he did was to take a frog's heart, this is a frog's heart, put it on a pole, as you could see, measure the way it bumps, and he did that by adding the liquid in which another heart was embedded, not by electricity. And that liquid enough was sufficient to cause this brain to pulse, as you can see. Okay? Now, this is the experiment he did. He indeed won the Nobel Prize, but before that, he had to leave Marburg because they would not give him a professorship. He refused to baptize. He was Jewish. So he moved to Graz in Austria. All that I heard from a historian of science who was present in the conference in Marburg. And remember, Marburg celebrated the 100 years to the discovery, but they refused to give him a position. In Graz, he became a professor. And in 1936, Germany annexed Austria. And overnight, he became a German citizen who is Jewish, who was not allowed to work. So the police threw him to jail. And he was really concerned, not for his life. He was concerned that his students will not know what to do the next day. So he sent postcards. He sent postcards from jail, which we saw in the lecture, that told the students exactly which experiments to do. And then the German historian said that the German government was in great financial difficulties. This was in the late 30s. They were getting ready to conquer the world. Definitely they needed a lot of money. And they reached an agreement with him. They took the Nobel Prize money. They gave him a ticket to a boat to New York. and this is where he died. So that movie was taken in New York. So it became very relevant to the Holocaust Day, actually. And the Department of Neurobiology in the Life Sciences Institute at the Hebrew University is named after Otto Lowy. Okay, so what do we know today about cholinergic neurotransmission that we did not know that time? We know a lot. So there are genes that tell a born nerve cell, you shall be cholinergic. You will react to acetylcholine. And those are regulatory genes, and they are unique to particular brain regions. So the brain is a very complex organ. In each particular part, there are different genes that are expressed, and they dictate the identity of the nerve cells that develop there. And they create complexes with each other, but this is a detail. Now, what do we mean when we say cholinergic neurotransmission? We talk about the production of acetylcholine, which involves several enzymes. Then we talk about packaging of this acetylcholine in vesicles, which are those little particles within a nerve cell that will fuse with the membrane, open up, and throw the neurotransmitter, the chemical, out. Okay? So here are the vesicles. It's thrown out, and when it's out, it will then look for and interact with another set of molecules, the receptors. When the neurotransmitter interacts with the receptor, they open a channel and ions can flow in, and this is what activates neurotransmission. Is that all? Not at all. You need to degrade it. Every communication needs to end. Sometimes to end the communication is not less important than to initiate it. So we have enzymes that degrade acetylcholine. And then we have proteins that can block the receptors and prevent them from interacting. And just to make it even more complicated, acetylcholine is not only a neurotransmitter, but it's a messenger that tells immune cells, stop inflammation, control disease. And to add up on all of that, we have microRNAs that control this system, which I hope we will get to presently. So we worked for years on the cholinesterase genes as the subset of genes that reflect cholinergic neurotransmission, and we'll talk a little bit about that too in the future. Okay. There is an issue about Alzheimer's disease. 95% of the patients develop it without inheritance of a mutation. What do I mean by that? A person who develops cystic fibrosis is sick because they carry a mutation in the cystic fibrosis gene. But most of the people with Alzheimer's disease do not have an Alzheimer's gene that is mutated. Some of them do, but this is very few. And those that do, they carry the mutation in a gene that is called amyloid, and that gene produces a neurotoxic peptide, A-beta, which is shown on the screen, and that peptide clumps together and causes those brown precipitates that Alzheimer's saw without knowing that it's a gene or a peptide or anything. And the main treatment that is used today, probably the only treatment that is used today, is to prevent the destruction of acetylcholine, to protect those remaining neurons in the brain and help delay the cognitive decline. And one of the 4 leading drugs in the world that are used to treat that was developed by Professor Marta Rosin in our medical school, in School of Pharmacology. She will light a lantern in Yom Ha'atzmaut. So anyone who's interested, you can see her. She's a new immigrant to Israel, 40 years ago, from England. Still, we have a big puzzle. What is the cause of the disease in those small fraction of 95% of the patients who don't have mutations in the amyloid gene? So today we know there is an amyloid gene. We know that several mutations in this gene may cause Alzheimer's disease. So there are families worldwide, in Germany and Italy and Russia and East America, that carry these mutations. And these poor people develop Alzheimer's disease at the age of 50 or so. Still there is no cure. Most of the other patients who don't carry one mutation that is recognized develop it at a later age, 60s, 70s, 80s. Okay. So how can we use genomic information to cure Alzheimer's disease? Yeah.
Yeah, I have a question first about the disease itself.
Okay.
So the problem is in the neurotransmitters or in the material?
The nerve cells that produce acetylcholine are the ones that die first in the disease, a whole population of them. This is why this was the mechanistic basis on which the currently available treatment was developed. Okay? But later on, other nerve cells die too. Okay? So how can genomic information be used to develop new drugs? And I'd like to tell you about an article that appeared 3 years ago in Nature, which reflects genetic studies in Iceland. Now, Iceland is a very small country. About 1,000 years ago, several hundred people were sent to Iceland from Sweden to, I think, fishing and perhaps just finding raw materials for Swedish purposes. And the whole of the Icelandic population, which is about 340,000 people, are the descendants of those few hundred families, which means in Scandinavia the genealogy is written very, very well. They have records for 1,000 years on who got married to who and which diseases were in the family and who died when. And it's the same people that still live there. The current Prime Minister of Iceland studied in the United States, in New York. Then he came back and he had apparently discussions with his friend from New York, and he realized that the genomic contents of his population are a research treasure because they know everything about the medical background, about the genetic background. The people are there. And the Icelandic government transferred a law saying that in Iceland the DNA is the property of the government. Just imagine. It's a real law. And they created a company called Decode Genetics, together, surprise, surprise, with that guy from New York. And they keep the records of everyone in the country, from clinics to genetics. And they published a remarkable article 3 years ago that said, we found a mutation in the amyloid gene that protects from Alzheimer's. The same gene where mutations can cause the disease is found in people who live over 100 years and they keep cognitively alert, and they carry a mutation that prevents the precipitation of the amyloid peptide. And I can assure you that today they are sitting and trying to find out small molecule drugs based on this concept that they discovered that will mimic the protective effect for the benefit of everyone. Now, does any of you know a drug that was developed based on genetic and structural information in another disease? So let me give you an example. HIV. AIDS was a fatal disease until about 20 years ago, I think, right? When the viral genes were sequenced, the proteins were crystallized, the structure was found, new molecule blockers of those viral proteins were developed. Today, AIDS is still a very serious disease, but it's a chronic disease. It's not fatal, because patients need to take a lot of drugs to block the viral proteins, but they can survive for many years. So what I'm telling you about the protection against Alzheimer is not just a dream. This is something that is happening today in labs worldwide. So could we say, like Schopenhauer did, that fate gives us the hand and we need to play the game as best we can? Perhaps so, but maybe we need to adapt it today to what we know about our genome. None of us carries the same genome. Even identical twins who do carry the same genome might have epigenetic regulations that change the expression of their genes. So the more we know about our genome, the better we would be protected against this and other diseases. So what are the other proteins that are expressed differently in Alzheimer's disease, and can one design drugs against those? So we would say that neurons have a special structure, right? There's a cell body, there are axons that are very long, there's a synapse that interacts with other neurons at the other end, there are dendrites that receive information. It's a very specific structure. That means that a nerve cell needs a cytoskeleton, a scaffold on which its structure is kept. And that cytoskeleton is called microtubules. So if you compare that in analogy to a building, these would be the metal elements that keep that cement building alive or stable. And the protein that stabilizes those microtubules is called tau. And in Alzheimer's disease, there are changes in the tau protein. So not only there's precipitates outside the cell, but the nerve cell inside shows changes in its cytoskeleton. It creates what we call tangles, neurofibrillary tangles. And actually, Alzheimer found both, the original Alzheimer. So this is how it looks. This is a plaque, precipitate, a brown precipitate of the amyloid protein. These are tangles. The black lines over here are the changes that are caused in the tubulin-associated proteins. Then there are other cells. These are nuclei of the other cells around it. And of course, you'll have this presentation on the website of the course, so anyone who wants to get into it deeper, you'll be able to. So Alzheimer saw plaques and tangles. And the question is, can we do something to organize this? So I have news for you. We don't agree. There are researchers in the Alzheimer field who we call Taoists. They believe in Tau. There are researchers who we call Baptists. They believe in the amyloid theory. And in each conference, there will be Taoists and Baptists, and they keep arguing until today. I just came back 2 weeks ago from Nice, where there was the Alzheimer conference of this year, and they were still arguing. So the people who believe in the amyloid, they say, this is the bad guy. This is what we need to prevent. The people who believe in Tau say, it doesn't matter what happens outside the cell. What happens is what happens inside. That's what matters. And what happens to the tau protein is binding of a lot of phosphate atoms, hyperphosphorylation. And this changes its charge, it changes its interaction with other proteins, and as you could see, it creates tangles in the cells. Is that all? Of course not. In addition to all that, there are modulator genes. So as you see, we know a lot, but we didn't solve the problem yet. What are the modulator genes? So this goes back again to the 60s in the last century, where some people were found to be at very high risk for heart attacks. And then their genotype was tested, and it was found that they are so sensitive because their lipid metabolism is impaired, and this is because of another gene, apolipoprotein E. And in the apolipoprotein E, we have 4 different variants. And any of us might have any dual combination of these variants, either 1 and 1 or 4 and 4. And what was found is that apolipoprotein 4 is a bad lipid metabolism controller. What you see here is that 4 is an open structure as compared to the 3, which is a closed structure. And all this is because of one amino acid that is modified. This is enough to change lipid metabolism. And the people who were at high risk were developing heart attacks and dying. But then cardiology developed. So open heart surgery was developed, stents were developed, a lot of statins were developed to reduce lipid in the blood. And those people at risk started to live longer. We did say life expectancy is considerably longer. They reached the age of 80, and then it was found that they are at high risk for Alzheimer's disease, which tells us something else. Lipid metabolism is also important, and changes in lipid metabolism may make you more susceptible to this disease. So how can we fight this? Immunologists believe that we can fight Alzheimer's disease by boosting the immune system. Immunologists believe that any issue can be solved with boosting the immune system. They might be right. So there are different approaches to try to tackle this. The first one, this is a company that says all we need to do is boost your immune system, give you more immunoglobulins, and then the brain will take care of itself. That's one approach. So far, no great success. But then people said, if we know which are the proteins that are the bad guys, maybe we should develop antibodies against those bad guys. For example, uterine cancer is caused by a papilloma virus that was developed decades ago, and now there is an immunization, and it solves the issue. So why not do that against Alzheimer's disease? Immune people before they develop plaques, and then we'll have a solution. So the very first person who suggested that in the wide world is a professor from Tel Aviv University. And she developed antibodies, and she showed that they precipitate the amyloid protein, and she wrote a patent. And then she was invited to be a consultant of a company called Elan that wanted to develop that approach. And then Elan developed an antibody, injected it to mice that carry the human mutated amyloid gene and develop plaques in the brain, and could show that they clear the brain. And they wrote a patent. And then Tel Aviv University had a very smart lawyer who sued them. And he said, we had a patent before you. You hired our expert as a consultant, so you acknowledge this. And eventually, Elan paid Tel Aviv University $10 million. So apparently, they recognized this. Based on those findings, which were a whole lot of noise around that, they got approval to start clinical trials. Now, clinical trials have 3 phases. The first phase is a safety phase. All you need to do is prove that your drug is not dangerous to treat people. You can, in some cases, get special approval and try at that phase already to prove efficacy, that is, show some help to the people who are treated. Second phase is efficacy par excellence. And third phase is expansion, after which you receive an approval. So the people from Elan got an approval to start developing clinical trials. They started. 6 people died from encephalitis. So their brain reacted very badly to the antibodies, at which stage the program was closed. FDA forbids continuation of clinical trials because of that. Yeah.
What, can you please explain?
Okay, encephalitis is an edema of the brain which can kill you. okay, דלקת קרום המוח. 6 out of the people who got treated died. So that's enough for the FDA to stop the clinical trial.
6 out of how many?
I don't remember. But it couldn't be many because it was phase 1. They're careful.
I think I got a little bit lost
Okay.
before that. The antibodies that were created were for the proteins, for the genes. I got a little bit lost.
The antibodies Ilan tried were targeted against the amyloid protein that creates the plaques. Okay? And here are the original drawings by Alzheimer. I mean, he was a really talented artist. Just imagine that he had microscopes that were so poor compared to what we have. This is how an immunoglobulin looks like. And it targets an antigen. Our antigen in this case is the amyloid protein. So a lot of wise people went back to the drawing board and they said, this is important. Because if you can treat mice and the antibody gets into the brain and clears it, then it should be possible to treat people too, except that the antibody we used was unsafe. So I have a friend in Switzerland, in Zurich, who started developing better antibodies, and he did that based on the human immune system. Other people use mouse cells to produce antibodies. And he not only used human blood cells, but he selected blood cells from people who live 100 years and are protected. They probably carry the Icelandic mutation, but that was not even known when he started that. Now, the other question is, who are the volunteers that should be tested? And the question relates to what I told you in the beginning of this lesson. Suppose that 40% of the people who carry plaques in the brain don't even show a cognitive decline. If you want to prove that a drug works and all you will do is measure cognition, then you need to measure it in people who show cognitive decline. But if you design an antibody against the amyloid protein, you need to look at the amyloid precipitates in the brain. So another group in Pittsburgh developed a radioactive tracer that you can inject, put the person in a PET machine, positron emission tomography, and look at the precipitates of amyloid. This is done routinely almost today in the United States, although it's a very expensive test. And the Ministry of Health in Israel does not permit even importing these machines, because they say this will increase the health cost tremendously. And as long as there is no treatment, why should anyone know if they have or not have precipitates in the brain? But now what happened was a combination that I learned about in the meeting in Nice 2 weeks ago, and some people might have read about it in the internet or in newspapers. And this is a combination between the Roger Nitsch approach to prepare better antibodies and a company that bought from him the license for those human-derived antibodies and decided to test them on a smartly selected human cohort. What they did was to run each volunteer to their clinical trial through the PET scan. So this is something that couldn't be done in Israel, for example, and ran a cognitive questionnaire. So in essence, they doubled the prospects of seeing a successful result to their test based on the properties of those patients. This is Roger Nitsch. So if you see him in the street, you'll know. This is the way people make antibodies today. So they grow mice, they challenge them with an antigen, they expose them to an antigen. So in our case, they would be exposed to precipitates of amyloid. Then they harvest their spleen cells, they grow them in culture and prepare immortal cell lines. Those cell lines are producing immunoglobulins, and from those you purify the antibodies that are immunoglobulins. What Roger Nitsch does is doing that in human-derived cells or biopsies, which is much more, excuse me, much more expensive approach. Combine that with a much more expensive approach of screening the volunteers. The company invested a lot. They paid Roger, that was the rumor in the corridor, so don't take me for that, $200 million for the license. And they ran a phase 1B trial, 1B meaning it's still phase 1, it's still looking for safety, but it also looks at the potential efficacy. 130 patients. This is unusual. Usually phase 1 is 10 to 20. Why did they do that? I can bet that they promised to pay him more when they reached phase 2, and they didn't want to reach phase 2 yet. But anyway, what was reported in that conference by Roger Nitsch is that that antibody reduced the decline in cognitive capacity of the treated patients. It was tested for 2.5 years, while reducing the amyloid load. So it seems that we have a real new hope for an immune treatment, which is problematic news for the Israeli Ministry of Health that will probably have to allow now to introduce these machines and add them to local hospitals, which indeed will raise the health costs considerably, but may now be justified. Okay. All this comes to say that we need to look at the genome of people, and we need to know about their cognition at the same time. And there is a very strong consortium that looks into those data, and it's called Enigma. Anyone knows what Enigma means? Of course. So this is what Enigma means. Okay, so I keep bringing you slides from movies, and again, this says that the word enigma carries a sociological weight today. So Alan Turing was the initiator of the machine that broke the Enigma code of the Germans. Again, I really didn't mean to prepare this for the day of Holocaust, but it's meaningful anyway. And anyone who didn't see at the movie, it's highly recommended. And the papers written by Alan Turing about the concept of his machine are released very slowly now. 2 years ago, the government in England released several of his papers from the 40s of the last century. Imagine, it's still considered a secret. Now, why do I bring that up? Because the Enigma consortium combines genomic information with brain mapping information. And I think that this is our day. So this quotation is from Winnie the Pooh, of course. And he asked Piglet, what day is it? And Piglet says, it's today. And Winnie the Pooh says, this is my day. So I feel sometimes that way. This is the day of neuroscience to combine the wealth of information and tools that we have today into a new know-how. How do we do that? We can today take the extracts from brain or the DNA and run sequencing. We can find out how does our genome look and what are the products of those genes in a particular tissue. And what the system does is to break everything to little pieces, sequence it rapidly and effectively, and then the computer looks for adjustments to the genome, which you see here. So this is the genome. There are tiny bits and pieces. A lot of information is carried out, and this is analyzed to tell us which genes exist in a system and what products do they make. The Enigma consortium looks at the genes expressed and the composition of the genome in 3 cohorts. By the way, there are 128 countries that are members in the Enigma consortium. We are not part of them, probably because there are insufficient numbers of patients. because they look at huge cohorts where they have both brain scans and genomic information. And what they did was to look at individuals, again volunteers, from a group of controls, minor cognitive impairment, these people who don't remember where they left the keys and where, and so on, and Alzheimer's disease. The people had a magnetic resonance imaging scan, MRI, and their genomic composition was subjected to what we call genome-wide association scan. So the entire genomic information is available with many, many different markers that say this is the way the genome looks.
What is it,
MCI, minor cognitive impairment. 80% of MCI patients are at high risk to develop Alzheimer's disease. Okay. The association scan is based on over 500,000 flags across the genome that say this person carries this flag and that flag and that flag. Together, it gives a good picture of one's genome. By the way, there are companies who do genome-wide association scan and give you some information, and they were forbidden in the United States because it's of no value. The analysis was apparently inappropriately done. But this was a serious consortium. They spent, I think, 2 years on automating the procedures of analysis to make them comparable in different centers. This is how the chips look. You put a lot of pieces of DNA on something that looks like a microscope slide. You hybridize it with probes, and you get a list of those 500,000 markers of sequence changes. We call them single nucleotide polymorphisms to dissociate them from mutations. These are not mutations. They exist in over 1% of the people at any time. So there are SNPs, single nucleotide polymorphisms. Each of us carries a different composition of SNPs. And what the Enigma Consortium started publishing is there are apparently SNPs that associate with different rates of changes in hippocampal volume. So you see what they did. The MRI scan gives you a section of the brain, and then you can find the hippocampus, which I showed you before, and just measure the volume. Measure the contours of the hippocampus. The genome-wide association scan provides you 500,000 entities, which says these are the nucleotides at this and this and that location. And then you look for people out of 10,000 who carry a marker that associates with a change in brain volume. And what they find is 5 different markers in different genes, and these are called the risk alleles. Each allele increases the rate of hippocampal aging by 4 years. So if you are unlucky and carry 5 of these alleles, your hippocampus will age 20 years faster than the other person. And that's why some people reach the age of 100 and remember everything, and others don't know where they left the keys at the age of 50. So this is the prediction right now. They continue the research on other elements. They already have one paper on IQ. There are alleles that associate with IQ, which is a very sensitive matter. So they release the papers slowly. Each paper starts with a page of the contributors, which is sometimes longer than the paper. And we call that, איך אומרים קבר אחים באנגלית? Because we remember the first author and the last one, but nobody else. Anyway. So this is a new way to do research which didn't exist until quite recently, but may come up with important information. So when we come to that, I'd like to remind you the quotation that says what a scientist is. And a scientist is someone who knows more and more about less and less. I'm afraid I belong to that group. And I have a confession to make. I was teaching for more than 20 years Introduction to Molecular Biology in the Life Sciences Institute. And what I taught is not true anymore. So I was telling students, a gene is a piece of DNA that makes RNA that makes protein, right? Wrong. There are genes that make RNA, but not protein. I was telling them 2% of our genome are genes, the rest is junk, right? Wrong. We didn't understand what the 98% do, but it's not that it doesn't do anything. So what we know today is that the nucleus includes a lot of genes that make RNA, and some of that RNA, a small part, leaves the nucleus as messenger RNA that makes protein. Other families of RNA are what we call now non-coding RNA. There are different families of non-coding RNA, and they do have jobs to do, but they don't make proteins. They're not coding. There would be some of these non-coding RNAs that will stay in the nucleus, others get into the cytoplasm, and all of that will determine what happens to the cell. Now, the messenger RNA gets out and a poly-A tail is added to it, which stabilizes it. And I was greatly relieved to see that this is still true, because that was my PhD thesis. So this one bit of information is correct. But what about the non-coding RNA? For example, microRNAs. They are 100-fold smaller than a regular gene. They can't do any protein. What they do instead is block other proteins from doing their job. And all that remains, with another quotation of Da Vinci, is to simplify this message, which is not a small matter. Okay. So let's try to simplify the case for microRNAs. Let's think about DNA that includes some genes that are microRNA genes, 100-fold smaller, but they're still genes. So they prepare RNA, and that RNA gets out of the nucleus, finds a complex of proteins that is waiting there as the machine in which the microRNA functions, finds a messenger RNA with a complementary sequence, binds it, and stops it from being active. We know about microRNAs less than 20 years. There was already a Nobel Prize given to these two people from California for discovering this new concept, because it really changes the way we understand how genes work. 95% of the research in the world on microRNAs involves cancer, because a lot of them change the way cells divide. We study microRNAs in the brain. So if you want an analogy, this is my analogy to how microRNAs work. A lot of traffic, and there are two little policemen in the center, and they guard it all. So these are microRNAs for you. One microRNA can control many genes, a whole pathway. One gene can be controlled by different microRNAs, which means that they are really complex, which makes it challenging, but even so, much more interesting. Yeah?
I didn't get what is their goal, the main goal of the microRNA?
The main goal is to wrap around those genes that show complementary sequence to them, stop them from being translated. Instead of doing something, they prevent something from being done. They block expression. Now think about that. You are a cell, and you want to be as effective as possible in an economical way. It makes much more sense to stop a whole pathway, like a dimmer of electricity, okay, than to start producing things to change things. Okay? Anyone who's interested, I wrote a popular science article about microRNAs in Hebrew, though. So anyone who reads Hebrew and wants to read that article, send me an email. I'll send you the paper. Okay? Because they are really a fascinating family of genes. So now, what is the importance of microRNAs? For many years we believe, scientists believe, that our brain is larger. You can see it is larger than that of other animals. More complex, more capable, can do many more things. And the question is, how did that happen? In evolutionary terms, this switch occurred very rapidly. So for years, scientists were saying, this is because of regulatory genes that change a whole pathway and therefore create an evolutionary change that is very rapid. And they thought about those genes that regulate transcription, because what they do is they bind a sequence that is complementary to them, and they allow a gene to be activated. Now that we know about microRNAs, why not them? It's the same, except that they are 100-fold smaller, much less energy to produce them. And the question is, if microRNAs were involved in evolutionary changes of the human brain, then we also have a problem. To create a rapid change in a species may kill its prospects to survive. So what you need is to adapt the change of the microRNA with the change in its target genes, in the pathways that it controls. And this was our prediction in this study that was done by Shahar Barbash in the lab. He's completing his PhD today. And the prediction said, brain evolution involved rapid changes in regulator genes. This is an axiom in the field. Everyone believes in that. MicroRNA target interactions could be related to this process. But if that is the case, then the interactions should happen together so that there'll be an adaptation of the changes in microRNAs and their targets. So this is a molecular evolution concept. And we checked it. How do you check that? And again, this relates to what I started with telling you, that there are new technologies. There is a website today that includes information that is called the 1,000 Genomes Project. And it includes the complete genome of many different people from different ethnic origin, different populations, different continents. Today it has over 1,200, so it's not 1,000 genomes anyway already. We also know the DNA sequence of the Neanderthal man. I keep showing here the Carmel Mountain because that's the most southern place in the world where the Neanderthal man lived. And if I talk about that overseas, there's always someone who says, but the Neanderthal sequence is ours. And they're right, that's okay. But still, we are the most southern place where the Neanderthal man lived. And of course, there is available on the web a lot of DNA genomes from different animals. So you could look at the genome of different species and ask, what about the microRNA genes? I know to identify them. Did they exist? When did they get born? And this is briefly what Shahar found. So what he did was to search in our human genome for microRNA genes that exist in our genome all the way through vertebrates. Of course, Darwin would be angry with us because he didn't know about the genome. Anyway, so there are microRNA genes that you find all the way back to all vertebrates. They are evolutionarily old, not too many of them. There are more microRNA genes that invaded the genome with the development of mammals. So they're younger. There are microRNA genes that are primate-specific. And some of them we didn't even find in the Neanderthal, although we don't have too many sequences of the Neanderthal, so we need to be careful about that. And most of them have targets in the brain. So altogether, what we found in that study is that our findings are compatible with the hypothesis that microRNAs were part of the evolution that changed our brain, and perhaps it's still changing. When my 3-year-old granddaughter looks in the newspaper and she wants to enlarge a picture, she does that. She doesn't realize that a static picture cannot be greater. Her brain is reacting differently from mine. And it could be that the effect of microRNAs on our brain is really changing the way our brain functions. So we prepared a cover page picture, which we sent to the journal, and the journal didn't accept. So now we are running another microRNA study just because we need to keep that picture. Okay. Now we can also look at the evolutionary conservation of microRNAs. And that started when I gave a lecture in Tel Aviv, and someone in the audience, a very smart scientist, asked me a question. So how would these genes evolve? They're too small. And I said, indeed, I think that the main evolutionary change would be in the targets that are much bigger. Statistically speaking, the prospects of them being subjected to mutation is much greater. So let's look at the genomic variation. We can check a microRNA gene and find out if it's stable or if it's subject to change. And we can change the target too. So this is the evolutionary conservation rate of microRNAs. You see that it's low to medium. This is the evolutionary change in the target genes, much wider. Target genes can go into high-level changes. And if we look at the Neanderthal man, then we would say that here we will have very stable microRNAs, highly flexible targets, and there is always a crosstalk between changes in microRNA genes and in their targets. So you asked, what does a microRNA do? This is what it does. This is an atomic resolution level model of one microRNA. That is the focus of Geula's PhD thesis. And this is its target, which happens to be the acetylcholinesterase gene that we are interested in. And you see that the orange part wraps around the green part and just blocks it. And because of that, it cannot be translated. It's evolution. It's exposed to destruction. Okay. So we look at microRNAs in the cholinergic system. What do I mean when I say we look? What does an experimental biologist do when we want to study the function of genes? And the answer is we make a mouse. So the microRNA 132 gene is targeting the acetylcholinesterase gene, and it is conserved. It exists in all mammals from the mouse on. What happens if we take off this region that talks to microRNAs and we insert the human acetylcholinesterase gene into the mouse? So now we'll have a mouse with much more acetylcholinesterase and no microRNA regulation. We killed it. What will that mouse do? So we made that mouse. And this is what these mice do. They run. This is a normal speed photography. They keep running. They're really nervous. If we put them into anxiety tests, they come up really anxious. They are 15% lighter than other mice, so you could say, this is good. Except that when we put them in a serial maze, where we put the mice here, and they need to learn, which they do very rapidly, that there's apple juice at the other end, okay? So a normal mouse does that very rapidly. This is the engineered mouse. See that? It goes back and forth. It's forgot its glasses. It doesn't know where it left the keys. It's doing retracing errors. This mouse is stupid. So it's both anxious and stupid. We didn't stress it. We just engineered it not to respond to this microRNA. Not only that, I did tell you that the cholinergic signaling controls inflammation. This mouse suffers from massive inflammation. Human patients with intestinal inflammation show huge increase in this microRNA. So we really proved the concept. This microRNA sends messages from the brain to the immune system. It controls both stress reactions and inflammation. And this is dependent on its interaction with its target, the acetylcholinesterase gene. So this is the study of the human patients. You know, people with massive intestinal bowel disease, they go through surgery. And the surgeons remove the inflamed tissue, and we got tiny little pieces of those tissues, and we check the inflamed tissue and control tissues from the edges, and you see that the inflammation associates with massive increase of this microRNA in human being. Okay. Which is a good thing, because microRNAs are what we call druggable. You can synthesize a DNA-based drug and block their function. So here is a gene where you don't need to look for a small molecule and try different chemistries. You need to put the sequence into a machine, develop a drug, and block it. And actually we have a drug like that, testing improvement in inflammatory bowel disease in BioLine RX, which is a Jerusalem incubator. Okay, so far, for a normal mouse, normal people, normal age, what about Alzheimer's disease? So I have news for you. Most of the microRNA genes in the Alzheimer brain go down. If this is the distribution of microRNAs, most of them are on the left side of this line. And one that is really changed drastically is 132. So now think about what I told you about treatment. Patients with Alzheimer's disease, I told you, lose most of the nerve cells that produce acetylcholine. Okay? And still, we give them drugs that block acetylcholinesterase, which is produced in exactly those cells. Why? Because we now know they lost also the microRNA that controls the production of that enzyme. So we ran that study together with many different researchers from many different countries. So you see a long list of contributors, but it was published, and I think it's believed by many. So is Alzheimer's disease the loss of one's capacity to react to stressors? Maybe. In many cases, in a stressed neuron, you see changes in gene expression, changes in proteins accumulation, changes in the structure of neurons, and this all involves the capacity to react to stressors. So how does this relate to the genes of the patients? We talked about acetylcholinesterase. This is how that protein looks like. It was crystallized in the Weizmann Institute by Yoel Zussman and Israel Silman, and it was found to act like a vacuum cleaner. It really shoves all the acetylcholine into a big groove and degrades it very rapidly. 10,000 molecules per second, so a lot. People who carry mutations in cholinesterase genes are very sensitive, for example, to insecticides, because insecticides are our chemical warfare agents. What they do is block the insect's acetylcholinesterase. And insects' fly muscles depend on acetylcholine. So when you see a small airplane spraying things on fields, don't go under it. It's unhealthy, because we have an acetylcholinesterase that is 50% identical to that of the fly. And people who carry mutations in cholinesterases are very sensitive to exposure to insecticides. We ran an analysis of electroencephalography. I should have stopped already? No. Okay, fine. Okay. We ran an analysis of brain mapping in carriers of such mutations several years ago, and we found that if these people just live next to sprayed orchards, they show hyperactivity in the frontal lobe. This is what happens under stress. They show down activity. The blue showed reduced activity in the deep nuclei, which reflects learning and memory impairments. So if you wish, this blockade mimics the reactions to too much microRNA. But at the same time, we knew there are mutations also in non-coding regions. And we thought we were wise. You know, that's not interesting because it couldn't do anything. So we knew about those mutations, we didn't study them. Now we know better. So could it be that those mutations in the regions we didn't seriously respect are involved in interaction with microRNAs? So that reminds me another hero, which is Eric Kandel, again a Nobel Prize from New York, who started from psychiatry and keeps saying that the future of psychiatry is only dependent on its combination of genome interaction. So, how could changes in gene affect brain functioning? Suppose we regard microRNAs as the Swiss knife of our brain. They have a lot of brain targets. They are changed with evolution. Each of us carries a genome. A small change in one binding site of one microRNA could make a difference. So the microRNAs don't function in a space, in an empty space. They function in the context of the genome of the person which expresses them. How can we measure that? We developed a technique which Geula ran, and this technique is called surface plasmon resonance. It involves tiny gold leaves that are put in a machine. You can bind to the gold leaf a sequence of a microRNA, and then you have microfluidic cells. You can stream across it a mimic of the gene that that microRNA is supposed to interact to at different concentrations, find out real dissociation constants. So I did promise you new technologies. Here is one. So now let's look at a working hypothesis. Suppose there is a microRNA. Let's name it 608. And it blocks acetylcholinesterase. You know, I worked on acetylcholinesterase many years. I published a lot of papers about it. And not too long ago, there was a dinner, festive dinner of the Hebrew University donors, and they put me the same table with a French clinician. And the guy says, "How do you spell your name?" He takes his BlackBerry. Oh, acetylcholinesterase. So that's how I spell my name. So this microRNA controls acetylcholinesterase. Now suppose we will have a mutation that will change that capacity. So what do we expect? What would happen? We would expect that we'll have too much acetylcholinesterase because this microRNA is not doing its job. But we'll also expect that we'll have a lot of molecules of the microRNA that are not employed now. So they will look for other targets. So our prediction would be elevated level of acetylcholinesterase and reduced level of the other targets. So this was our working hypothesis, and we wanted to check it on acetylcholinesterase. So that means we look for signals from body to brain, inflammation, anxiety, and so on. Okay. And when I say we look, this is Geula. You might recognize her, and that's her paper on this. And what Geula found is that carriers of a mutation that impairs this interaction, just one single nucleotide out of 4 billion, adenine to cytosine, have changes in anxiety. Their brain shows much more acetylcholinesterase. They show more acetylcholinesterase in the blood, so you can test that. And they appear in much, much higher incidence in black tribes from Africa in the GWAS collection compared to Caucasians, 34% in Africa, 4% in the United States. Now, someone should ask me, what about us? So, of course, we did us too. It's not published yet, but I can tell you that in Israel, we find 17% carriers. So, first thing, if you ever wondered whether the theory about migrating populations is correct from Africa through the Middle East to the West, it is correct. We are exactly in the middle. Second of all, we are an anxious tribe. And in connection again with the Holocaust, I talked several years ago to an expert on stress, Professor Sapolsky from Stanford. And he told me, I come from an anxious tribe. Anxious tribe, that's us. He said, I would not have stayed in Germany 1 week when they started, the Nazis started getting up. But here what we see is a much higher tendency for anxiety in black tribes compared to white ones. We also find higher blood pressure in carriers of this single nucleotide polymorphism. So these were healthy athletes, very young people, 34 years of age, normal weight, doing frequent athletics. And they should be really careful because their genome carries a marker that says that they may be subject to aging diseases. So what are the aging diseases they may be subject for? I'd like to show you what we measured in stroke patients. Stroke is a major risk factor for Alzheimer's disease. And we published, and again we is Dr. Shani Shenhar, who is a postdoc in the lab. We published an article 5 years ago that said stroke patients have much lower acetylcholinesterase activity in their blood. And those who have very low activity are at risk not to survive the year after, probably because their immune system doesn't function or overdoes. And those who showed no decrease or very little decrease suffer poor recovery. That is, after 2 or 3 years, they did not regain their consciousness. Now we know how the genes look. So we genotyped these patients, and we compared the data psychologically to what they were in admission and after 2 years. And what you see here is that carriers of this single nucleotide polymorphism, which is in a non-coding region, no one would take it seriously until we did this study, do not regain cognition 2 years after stroke, where other patients do. They do not regain their memory. They do not regain their capacity for executive function. And this could be because they have too much acetylcholinesterase, which is what we found before. But now it says maybe these people are really good candidates to be treated with the Alzheimer's disease drug that Martha Rosin developed, because if they have too much esterase, nobody treats stroke patients today with any cognitive drug, but perhaps this could be tailored to those carriers of this change in the genome. In other words, what we're talking about is personalized medicine. And we more and more these days say that treatment of patients is still done. You know, what's the basis of drug dosing? Can you think about how does a doctor calculate the amount of drug you are about to receive? Only weight. Not gender, although we realize that women are not equal to men. Not height, not anything else, only weight. And in 5, maybe 10 years, we'll come to the doctor in the clinic. He will have our genetic information on the website. And say, oh, it's you. You need 2 aspirins twice a day. So, okay. So what did we talk about? The message behind Geula's article said that excess of this microRNA would not only release a lot of acetylcholinesterase, but excessively suppress other targets. This is another target. And what it does is to change the vicious cycle between cholinergic signaling and anxiety and inflammation. Now we know that anxiety and inflammation talk to each other. Inflammation increases anxiety. When we are sick, we don't feel very well. Anxiety increases inflammation. Something needs to stop this vicious cycle. And what our study indicated is that perhaps some of these people who are known to be anxious, actually there are many more than those, would have an impairment of one of the microRNAs that control one of the genes that are involved in regulating anxiety and inflammation and the connection between them. So let's take one example. This is one microRNA that we studied. We know that it has many targets. We know that it controls inflammation, anxiety, and blood pressure. We found it. And we know that if we change the ratio between its capacity to block different targets, we elevate anxiety, inflammation, and blood pressure. So how do you study that? I told you already. What does a biologist do when they want to study a concept? No? Make a mouse. There are no mouse with microRNA 608 because it's a primate-specific microRNA. But Bettina Nadorp in the lab has engineered a mouse with the human microRNA. And we already know that it's expressed, the microRNA, in the same tissues as in human. We still don't know what is the phenotype because the mice are still growing. So, 5 last minutes. We want to look at the genes that are modified in Alzheimer's disease. So I brought you pictures from a famous artist. You can find this on the New York Times website. His name was William Utermohlen. He lived in New York, and he drew self-portraits and developed Alzheimer's disease. And as he was deteriorating at the time that the pathology spreads, he lost the capacity to see details, to draw space. It's a very famous example. What we've done in the lab is to look at those data sets that are available on the web. Now we have our own, but initially we didn't. And ask which parts of the nerve cells, which genes are modified and where are they active at the different stages of Alzheimer's disease. And what we found is that the first genes to be damaged are those that control synapse functioning and mitochondria. And anyone knows what do mitochondria do?
It's the middle of the cell.
It's the metabolism center of control. So mitochondria controls our energy. The brain uses 40% of the energy, the calories that we consume. The brain is a region that requires a lot of energy. And those changes might mean 2 things. It might mean that the brain gets anxious and works over time. It might mean that changes in the mitochondrial genes predict and maybe control the subsequent processes in the disease. The changes are medium level, but then they become, in the next phase of the disease, they become more pronounced, and now we see changes in the nucleus as well. And at the end stage, we see changes in all regions. And we can't really know which cells are affected. We always have to remember that at that level, the brain has already lost a lot of its nerve cells. So is it related to acetylcholinesterase? Of course, everything is related to acetylcholinesterase. What we find is that patients at the early stage of Alzheimer's disease show elevation in acetylcholinesterase in blood tests that associate with the precipitation of amyloid plaques in their brain. And I think that I will stop that just by telling you that what we do now is to study gene expression in brains of patients with or without cognitive decline, with or without neuropathology, so that we can differentiate between those changes that relate to the dementia to those changes that are caused by or maybe affect the neuropathology. It's been a pleasure teaching you. Thank you very much.